So our next speaker is Dr. Erik Ullian, Associate Professor in Residence in the Department of Ophthalmology at UCSF. He'll be our final speaker from the neural maturation cohort. And so we welcome Erik.
Thank you. Thanks to Alan for inviting me out. I'm going to be talking about work that David Rowich and I have been doing to look at the role of astrocytes in maturing neurons and conferring appropriate function in complex circuits. So the general question that David and I were interested in is, what is the extent and consequence of astrocyte heterogeneity throughout the CNS in rodents and human? And in particular, David had previously shown that astrocytes can be described as heterogeneous populations of neurons based on gene expression. And a lot of this heterogeneity is region-specific and comes about from the radial glial cell of origin that give rise to astrocytes in different brain regions, such as cortex, midbrain, and thalamus, for example. And this heterogeneity is similar to work, for example, that has come out of Jeff Macklis's lab describing regional astrocyte heterogeneity, and other groups have described similar levels of heterogeneity. We've worked with David to show that there's actually functional consequences of this heterogeneity. For example, in ventral astrocytes, there's particular genes such as SEMA3A that are highly expressed in the ventral astrocyte surrounding motor neurons. And if you remove this gene from the astrocytes, motor neurons actually die and lose specific types of synaptic connections. And more recently, we've worked with David to show that astrocytes actually, as has previously been shown, are heterogeneous, for example, in their potassium channel KR4.1 expression that's expressed differently in different brain regions. And if you actually selectively knock out this potassium channel from astrocytes that surround large, fast-spiking alpha motor neurons, these alpha motor neurons will actually shrink down and become morphologically indistinguishable from slow-spiking motor neurons, and functionally indistinguishable as well, such they'll become slow-spiking motor neurons. So this suggests that some of this heterogeneity of astrocyte gene expression is really important for preserving the functional complexity of neurons. And this happens despite these neurons maintaining their normal transcription factor complement that demarks large, fast-spiking alpha motor neurons, suggesting that there's cell non-autonomous roles, as Mark Kirshner alluded to earlier, that are really important for maintaining some of the heterogeneity of CNS circuit function. So one question that came up is there more granularity to this astrocyte heterogeneity? Is there more heterogeneity even within specific regions where we know that there are different types of astrocytes based on gene expression and function? And in particular, David and I and Omer, a postdoc in his lab, have been talking about whether there could be layering of astrocytes in the cortex. But one challenge to be able to actually assay this is if we could actually look at gene expression in cortical astrocytes while preserving the cortical architecture and the complex architecture of the CNS. So to get at this question, Omer Bayraktar, who is a postdoc who worked with David and me and was in David's lab and is now at the Sanger Institute, used existing technology to set up a system to be able to assay gene expression at the single cell level through various regions of the CNS. So he basically took off-the-shelf systems that are already existing. So he could take brain sections from mice, put them on glass slides, and use automated multiplexed smFISH and RNA-scope combined with immunohistochemistry such that he could get 4 channels for RNA detection and protein detection, and then used an automated high-resolution imaging system that would allow him to get single-cell resolution throughout the entire cortical sections, or brain sections, rather, and then developed a machine learning algorithm that would automatically detect cells and be able to sort of demark them and then count the number of spots within the cell bodies. And so he was able to take this data and use the Allen Brain Atlas database in order to put this data back into specific cortical and brain regions, and worked with Dan Geschwin's lab to set up t-SNE plots and look at cell classification based on gene expression. And he set up this first iteration of this throughput system, which he called LASTmap, for large tissue area spatial transcriptomic mapping, in David's lab at University of Cambridge. So as proof of concept that this would actually work, the first thing that Omer did was look at cortical neuron layering and see if this could automatically detect and describe the well-known transcription factor differences that demark upper, mid, and lower layer cortical neurons. And so he used this method, which is shown here. This is an entire heavy section of a brain that was taken at this cellular resolution using this spinning disk confocal, and used a variety of well-known transcription factors such as CUX2, RORB, and CTIP2. And you can see that the program can automatically distinguish between cell bodies and then surround them and count the number of spots within them. And for example, in this piece of cortical section, it can convert this information into graphs like this, where each spot is an automatically detected cell body, and the intensity of the red basically indicates the number of RNA spots that were counted. And as you can see, allowing the system to automatically go through, we can detect upper, mid, and lower layer neuronal markers. And I don't have time to go into this, but using this in collaboration with Dan, they were able to actually identify some putative novel populations of excitatory cortical neurons. So the question, of course, that we were originally interested in is whether there are different layers of astrocytes. So similar to how there are different layers of neurons, it's well established, could there also be different layers of astrocytes that demark upper layer versus mid or lower layer astrocytes in the cortex? And so Omer applied this technique to the astrocyte gene expression. And he did this initially by using barrel cortex and dissecting out upper layer astrocytes, or upper layer cortical tissue and lower layer cortical tissue, and isolating the astrocytes based on ALDH101 GFP expression, which is a fairly robust marker specifically for astrocytes, and doing RNA-seq of the bulk tissue. And so surprisingly, when he did that, I think he and I and David were both initially surprised at how many genes he discovered that seemed to be enriched in deep layer versus upper layer astrocytes compared to bulk tissue. So initially, there were about 160 genes that he found that seemed to be differentially expressed in these two populations, general populations of astrocytes. And just a few of them are shown here, with cordon-like 1 is upper layer astrocyte gene, and ID3 is a lower layer astrocyte gene. And the question is, of course, is this really true? Can we actually take these genes that he discovered with RNA-seq, and can we map them back to astrocytes within the cortex and actually confirm that they do actually demark different populations of astrocytes, or is this just simply an artifact of the dissociation or some other issue? In addition, we're very interested in knowing whether or not this putative layering is preserved throughout the cortex, or it's isolated or restricted to certain parts of the cortex. So when Omer took this technology and applied it to astrocytes, there are several things he had to do to perfect it. First, he had to find a marker that would allow him to automatically delineate astrocytes robustly, and GLAST mRNA detection turned out to be very good and allow reasonably good detection of astrocytes and allow the machine learning algorithm to automatically surround and detect astrocytes. He then needed to subtract non-astrocyte signals, so he used NuN and synaptotagment as methods to detect non-astrocytes, and he's looked at other cell types as well to dissect out signal from non-astrocyte cells. And using this method, it showed pretty robust filtering for single astrocytes that was able to subtract neuronal and other non-astrocyte signals. And when he does that and looks at the array of genes, the genes sort of fall into these general categories based on heat map. And so what he found is that, of course, most genes actually really are pan-astrocyte genes. They're expressed throughout the layers, and they're not really highly restricted to one particular layer of astrocytes. But there's an interesting subset of genes that seem to be enriched for more upper layers and enriched for more deeper layer genes. And you'll notice, of course, for most of these genes, it's not a clear demarcation of upper or lower layer expression. There seems to be these big, broad gradients of expression. I think this is going to be a general paradigm for astrocyte genes, that they're not going to be as restricted as neuronal genes. So here's one example of a gene that's enriched in upper layer astrocytes. So here is a series of images through a cortical section from layers 1 through 6, stained for NUN, for GLAST, for astrocytes, where you can see the mRNA within the astrocyte population, and for this cordon-like 1 gene. And you can see that its expression seems to be higher in upper layers, and this is now seen here at the single cell resolution, where the program will automatically detect the astrocyte and surround it and quantify the number of spots within it. And these upper layer astrocyte genes express more of this cordon-like 1 mRNA, whereas the lower layer astrocyte genes appear to have fewer spots in them. And when we convert this into this similar single cell heat map, you can sort of see the distribution of these astrocytes that have been identified with cordon-like 1 gene expression. And again, it's a fairly broad expression pattern, although it is more heavily localized to these upper layers within the cortex in these astrocytes. So here now are several genes that sort of fall into the category of more upper, mid, and lower, and then white matter-like astrocytes. So again, cordon-like 1 is the one that I just showed you. There's also a very interesting gene that has come out of this called selin, which seems to be more enriched in mid-layer, layer 4 astrocytes surrounding layer 4 neurons. There's also genes such as IL-33, which at least initially, these are from P14, I should say, is enriched in lower layer astrocytes. And then not surprisingly, there's clear transcription factors that seem to demark white matter-like astrocytes, which are in layer 1 and layer 6 and below. So in general, there does seem to be some layering of astrocyte genes, which is something that we wondered about, and we were unsure whether such heterogeneity could actually exist within a single region. Now, again, as I mentioned, if you compare in these interpolation plots of heat maps, if you compare the neuronal layering genes, which are fairly precise and demark clear layers, with the astrocyte layer genes, you see immediately that the astrocyte genes are much broader, and that it does not exactly mirror the neuronal layering within the cortex. So it's reflective but different. So apparently the astrocytes may be following different rules from the neurons in terms of how they set up differential gene expression within these cortical areas. I think perhaps one of the most interesting things that's come out of this system for looking at gene expression across cortical areas is that we can look to see whether these patterns of expression are preserved in multiple cortical regions or whether they're restricted to certain regions. And you can see here, for example, if we compare the selin spots and look at the single astrocyte heat maps within these cortical regions compared with cordon-like 1, that for the selin gene, we see that it's highly expressed in somatosensory and barofield cortex, but actually its expression goes down quite a bit as we move into areas even visual and auditory cortex and retrosplenial cortex. So the patterns of expression are actually more complex than we initially realized. Some genes seem to be localized specifically into cortical areas, and interestingly, this particular gene follows the pattern of layer 4 neuron expression quite closely, whereas other astrocyte genes seem to be much more broadly expressed throughout the cortex. And of course, finally, we want to know whether this type of heterogeneity is preserved in human astrocytes, and there's actually reason to believe that human astrocytes might be even more complex, as has been mentioned in several of the talks earlier, both morphologically and by gene expression. And Omer has been able to apply this technique to postmortem tissue. So here, for example, is an example of human cingulate cortex from a 24-year-old used stained for cordon-like 1 with the RNA scope technique. And you can see that he's able to actually detect differences in expression based on the RNA scope spot counting, excuse me. So we're working both with David and Omer, both to understand the functional consequences in mouse and in human of these differences in gene expression. For example, by using, as shown here, iPS-derived human astrocytes seen with membrane GFP in the presence of neurons, for example. And we've recently started collaborating with Martin to be able to actually specifically manipulate gene expression in these cells using his CRISPR-I and CRISPR-A techniques so that we can actually start to interrogate some of the human-specific genes and some of the regionally differentially expressed genes to begin to uncover some of their function. So, in conclusion, this first generation of spatial transcriptomics that Omer has developed looks like it's going to be very useful. Right now, it's limited to 4 channels, but he's working on methods to actually greatly expand the number of channels that he can assay at one time. He certainly has provided evidence that there's layering of astrocytes by these very broad layers within the astrocytes, and that these layers diverge from neural layers. And we believe that the astrocyte and neuron layers are going to define complex and interesting parts of cortical architecture and function that we are in the process of pursuing. And the functional consequences are basically the next big question that we're interested in uncovering. And again, I'd like to thank the Paul Allen Frontiers Group for supporting this initial work and additional funding that we've received since then as well. Thank you.
Questions? Jeff?
I'm wondering for a few reasons whether you've expanded the temporal course of analysis earlier in particular. And I ask for a few reasons. One is, one could imagine that because the neurons born in those sequential layers span over about a week and are doing different functional things, that insofar as astroglia are talking to them, they might swing through. And then looking, when you pointed out that the layers aren't coincident, it almost looked like the astroglial genes were distributions that were waving, sort of sweeping up through the layers. And we found that very useful to have 3 or 4 critical times in the neuronal.
Yeah. So that's a great question. So we've looked at some older time points, and interesting, some of the layers seem to be preserved, for example, at P56, and some of the genes seem to disappear. They just completely disappear, like the selin gene. And we're now going back to earlier time points to try to understand what how this sort of develops. Simon Hippenmeyer, has done lineage tracing and claims that, and I believe this, that astrocytes are not born in a layer-specific manner, that it's very stochastic. And recently, Omer's done some experiments, for example, in reeler mice, where he's inverted cortical layers, and that seems to actually invert these astrocyte layers. So we believe the astrocyte layers may not be an intrinsic component of the astrocyte, but in response to signals from the neurons that we don't understand at this point. But we think there's this complex interplay between the astrocytes and the neurons back to the neurons that's basically essential for maintaining appropriate cortical function and regional identity.
Question? No, a microphone's coming too.
Erik, that was really nice. When you did the automated feature selection and you were able to trace out, did you notice any volume association or?
Yes. So there absolutely is differences in volume between upper layer and lower layer astrocytes. We normalized all the data based on volume to make sure that this wasn't artifacts of cell size based on layering and GLAST expression and things like that. So these layers are independent of astrocyte volume and amount of GLAST expression, mRNA expression, but there clearly are differences in astrocyte morphology among layers as well. Yeah.
And one last question over here.
Quick question. Have you done any work with mass spec to try to understand how well these transcripts predict protein expression?
Yeah, so we are working on trying to verify protein expression currently. And at this point, we don't know.
Okay. Thank you so much.